In the name of Allah, who is the most gracious and most beneficent. Assalamu alaikum and a very good morning to all of you. Well, my name is Bisma Arshad and I'm from the Department of Biotechnology. And today the topic of my discussion would be alfalfa in the field of chemistry. So basically, first of all, I'm going to tell you the contents of my discussion. Introduction, chemical composition, role of proteins, phytochemicals present in alfalfa, industrial applications, environmental impact, future prospects and challenges, utilization, extraction, commercialization of alfalfa, extraction analysis, analytical techniques, nutritional and medicinal value, potential health promoting properties, therapeutic applications and traditional uses in herbal medicine. Well, first of all, I'm going to tell you the introduction and historical background of alfalfa. Overview of alfalfa and its significance in agriculture and food production, historical background and cultivation practices. While there is a diagram which shows the plant and trees of alfalfa. While alfalfa, scientifically known as Medicago sativa, is a perennial flowering plant that is widely cultivated as a forage crop. It has been grown for centuries due to its high nutritional value and versatility in various agriculture systems. Alfalfa is known for its ability to fix nitrogen from the atmosphere, enriching the soil and making it a valuable component of crop rotation systems. Well, to be continued. To be continued here. Here's a diagram where you can see the breeding alfalfa. Historically, alfalfa has a rich background dating back into ancient times. It is believed to have originated in Central Asia and was cultivated in ancient Persia, modern day Iran, around 200 BC. From there, it spread to other regions such as Greece, Rome, and eventually to Europe and Americans through trade and colonization. Well, alfalfa is valued for its versatility and numerous benefits in agriculture. It is primarily grown as a forage crop, providing high quality feed or livestock such as horses, cattle, goats, and sheep. The plant's deep root system enables it to access nutrients from the soil, making it highly nutritious for animals. Alfalfa hay, which is the dried and harvested plant material, is a staple in livestock diet, supplying essential proteins, carbohydrates, and minerals. Well, additionally, alfalfa is known for its nitrogen fixing abilities. Like other legumes, it forms a symbiotic relationship with nitrogen fixing bacteria in its root nodules. This enables it to convert the atmospheric nitrogen into a usable form, enriching the soil with nitrogen and reducing the need for synthetic fertilizers. This quality makes alfalfa an excellent rotational crop, improving soil fertility for subsequent crops. Cultivation practices for alfalfa involve selecting appropriate soil types with good drainage and a neutral pH. It requires full sun exposure for optimal growth. Alfalfa is typically established from seeds and can be sown directly into the ground or started in nurseries or transplanted. The plant is capable of withstanding harsh environmental conditions such as drought, making it a resilient crop in many regions. Well, when alfalfa reaches its flowering stage, it exhibits beautiful purple flowers that attract pollinators such as bees, aiding in pollination and supporting biodiversity. In addition to its agricultural significance, alfalfa has also application in herbal medicine and as a cover crop to prevent soil erosion. Overall, alfalfa plays a crucial role in agriculture and food production by providing nutritious feed for livestock, improving soil fertility, and promoting sustainable farming practices. Its historical background and cultivation practices demonstrate its enduring value and widespread use in various parts of the world. Well, now comes to the second topic, which is chemical composition, while starting from the macronutrients. Proteins. First of all, alfalfa is known for its high protein content with levels ranging from 15 to 25%. These proteins are rich in essential amino acids, making alfalfa a valuable source of dietary protein for livestock and humans. Carbohydrates. Alfalfa contains carbohydrates such as cellulose, hemicellulose, and soluble sugars. These carbohydrates provide energy and serve as structural components in the plant. Lipids. Alfalfa seeds contain lipids mainly in the form of oils. These oils are rich in essential fatty acids, including linoleic acid and alpha linoleic acid, which are important for human nutrition. Vitamins. Alfalfa is a good source of vitamins, particularly vitamin K and vitamin C. Vitamin K is essential for the blood clotting and bone health, while vitamin C acts as antioxidant and supports the immune system. Minerals. Alfalfa contains various minerals including calcium, magnesium, potassium, phosphorus, and trace elements like iron, zinc, and copper. These minerals play a vital roles in bone health, muscle function, enzyme activity, and overall psychological processes. While here you can see the picture like previous I talked about the purple flowers, so there it is. Phytochemicals. Alfalfa is rich in phytochemicals, which are bioactive compounds that contribute to its medicinal properties and health benefits. Some notable phytochemicals found in alfalfa include isoflavones. These compounds have estrogenic activity and may offer potential health benefits such as reducing menopausal symptoms and protecting against certain cancers. Saponins. Saponins have been associated with the cholesterol lowering effects and may possess anti cancer and anti inflammatory properties. Flavonoids. Alfalfa contain various flavonoids such as quercetin and camphophyrol, which exhibit antioxidant and anti inflammatory activities. Now I'm going towards the industrial application where we can include pharmaceuticals, alfalfa derived compounds, particularly as phytoestrogens such as isoflavones, have gained attention for their potential health benefits and therapeutic applications. Isoflavones such as genistein and diazine exhibit estrogenic properties and have been studied for their potential role in the hormone replacement therapy, osteoporosis prevention, and cancer treatment. Additionally, alpha extract has demonstrated anti-inflammatory and antioxidant activities, which are valuable properties in the pharmaceutical development. Here you can see a picture where there is a method of separation and determination. Now, cosmetics. Alfalfa derived compounds have found application in the cosmetic industry due to their antioxidant and skin protective properties. Extract rich flavonoids, saponins, and phytoesterols from alfalfa have been incorporated into skincare products to promote skin health, protect against UV radiation damage, and reduce the signs of aging. These compounds help to maintain skin elasticity, improve collagen synthesis, and inhibit the formation of the free radicals. Nutraceuticals. Alfalfa is rich in essential nutrients, vitamins A, C, E, and K, minerals, calcium, iron, potassium, and dietary fibers. As a result, alfalfa-derived compounds are used in the production of nutraceuticals, which are natural health products with potential therapeutic benefits. Alfalfa supplements and extracts are often consumed to support overall health, boost immune function, and enhance digestion, and provide nutritional supplementation. Now, environmental impact. Here you can see the picture where you can see how environmental factors affect them. Nitrogen fixation. Alfalfa is a legume plant that forms a symbiotic relationship with nitrogen fixing bacteria called rhizobia. These bacteria reside in nodules on the plant roots and convert atmospheric nitrogen into a form that plants can utilize. This process, known as nitrogen fixation, allows alfalfa to acquire nitrogen without relying heavily on synthetic fertilizers. As a result, alfalfa cultivation reduces the need for nitrogen fertilizer inputs, which helps to minimize nitrogen runoff and its associated environmental pollution. While now soil improvement, alfalfa has a deep root system that can extend several meters into the soil. These extensive roots help improve soil structure by enhancing water infiltration, reducing erosion, and increasing soil organic matter content. Alfalfa deep roots can also facilitate nutrient cycling as they can access nutrients from deeper soil layers and bring them to the surface through leaf litter and root decay. Consequently, alfalfa cultivation contributes to soil fertility, enhances soil health, and supports sustainable land management. While crop rotation, alfalfa is frequently used as a part of the crop rotation systems. Rotating alfalfa with other crop provides several benefits. Firstly, alfalfa deep roots help break up the compacted soil, allowing for improved root penetration and water movement for subsequent crops. Secondly, the nitrogen fixing capabilities of alfalfa leave behind residual nitrogen in the soil, benefiting subsequent crops that follow in the rotation. This reduces the need for the additional nitrogen inputs for subsequent crops, reducing environmental impacts associated with the synthetic fertilizers. Cover crop. Alfalfa is also employed as a cover crop primarily during fallow periods or between the cash crops. As a cover crop, alfalfa provides ground cover that protects the soil from erosion caused by the wind and water. It helps prevent nutrient leaching and weed growth, reduces soil moisture evaporation, and improves soil organic matter content. The dense root system of alfalfa helps retain nutrients and prevents them from leaching into groundwater or surface water bodies. Future prospect intelligence identification of the novel bioactive compounds. Despite the extensive research on alfalfa derived compounds, there may still be undiscovered bioactive compounds with unique properties and potential applications. Identifying and characterizing these compounds can expand the range of potential uses in pharmaceuticals, cosmetics, and nutraceuticals. Understanding the mechanism of action, further research is needed to depend our understanding of the mechanism of action of alfalfa compounds. Investigating how these compounds interact with biological targets and exploring their signaling pathways can provide insights into their therapeutic potential and help optimize their applications. Synergistic effects and formulation optimization. Studying the synergistic effects of different alfalfa compounds and their combinations with other natural or synthetic ingredients can lead to the development of optimized formulations. By understanding the interaction between the bioactive compounds, researchers can enhance the efficacy and stability of the alfalfa based products. Bioavailability and delivery systems. Investigating the bioavailability of alfalfa derived compounds and developing efficient delivery systems can improve their absorption and targeted delivery to specific tissues or cells. This can increase their therapeutic efficacy and reduce dosage requirements. Despite the potential, there are challenges in maximizing the extraction, utilization, and commercialization of the alfalfa compounds. Standardization and quality control. Ensuring consistent quality and standardization of alfalfa extracts and compounds poses a challenge due to the natural variations in the plant chemistry, growing conditions, and extraction methods. Establishing standardized protocols and quality control measures is crucial to guarantee the reliability and reproducibility of the alfalfa dry products. Extraction efficiency and scalability. Maximizing extraction efficiency while maintaining the integrity and activity of alfalfa compounds is a challenge. Developing extraction methods that are cost effective, environmentally friendly, and scalable for large scale production is essential for the commercial viability. Regulatory considerations. Alfalfa derived compounds intended for pharmaceutical, cosmetic, or nutraceutical application need to meet regulatory requirements for safety, efficacy, and quality. Navigating the regulatory landscape can be complex and time consuming, requiring sustained investment in research, clinical trials, and regulatory compliance. Market acceptance and consumer awareness. Expanding the market acceptance and raising consumer awareness about the benefits of the alfalfa derived compounds is crucial for their commercial success. Education and promotion campaigns are necessary to inform the consumers about the potential applications and advantages of these compounds. Well, extraction and analysis. Solvent extraction involves using organic solvents such as ethanol, methanol, or water, organic solvent mixtures to resolve the desired compounds from the plant material. This method is versatile and widely used for extracting a wide range of compounds from alfalfa, including phenolic compounds, flavonoids, saponins, alkaloids. The extraction process can be performed using techniques like maceration, reflux, or sanication, depending on the desired extraction efficacy. Supercritical fluid extraction. Supercritical fluid extraction utilizes a supercritical fluid, typically carbon dioxide, as extraction solvent. Under specific temperature and pressure conditions, carbon dioxide reaches a supercritical state with properties of both gas and liquid. Supercritical fluid extraction is particularly useful for extracting heat-sensitive compounds as it provides the general extraction conditions. It is commonly employed to extract essential oils and volatile compounds from alfalfa. Microwave assisted extraction utilizes microwave energy to facilitate the extraction process. The plant material is fixed with a suitable solvent and microwave energy is applied to induce rapid heating and facilitate the release of the target compound. MAE offers advantages such as reduced extraction time, improved extraction efficiency, and lower solvent consumption. It is used to extract various bioactive compounds from alfalfa, including phenolic compounds and flavonoids. Enzyme assisted extraction involves the use of enzyme to enhance the extraction process. Enzymes can break down the cell walls, degrade unwanted components, and release the target compounds from the plant material. Commonly used enzymes include cellulases, pectinases, and proteases. EAE can improve the extraction efficacy and selectivity of the certain compounds, particularly polysaccharides and proteins from alfalfa. While analytical techniques. These techniques include chromatography, spectroscopy to identify and quantify the chemical components. Here you can see the analysis of BHP. Chromatography separates and analyzes the components of a mixture based on their differential interactions with the stationary phase and the mobile phase. Two widely used chromatographic techniques for alfalfa analysis are high performance liquid chromatography HPLC employs a liquid mobile phase and a stationary phase packed in a column. It is particularly useful for separating and quantifying various classes of compounds in alfalfa, such as phenolic compounds, flavonoids, alkaloids, and organic acids. Different detection methods, such as UV vis, diode array detection, and the fluorescence, can be used in the conjunction of the HPLC to identify and quantify specific compounds. Gas chromatography utilizes a gaseous mobile phase and a capillary column to separate volatile and semi-volatile compounds. While it is commonly employed for analyzing essential oils and volatile compounds in alfalfa, it is coupled with detectors like flame ionization detector or mass spectrometry to identify and quantify the separated compounds. Spectroscopy techniques analyze the interaction between the electromagnetic radiation and matter to identify and quantify chemical components. In alfalfa analysis, the following spectroscopic techniques are commonly used. UV visible spectroscopy measures the absorption of light in the ultraviolet and visible regions by chemical compounds. It is useful for the determining and the presence and the concentration of the compounds that absorb in these regions, such as phenolic compounds and flavonoids. Fourier transform infrared spectroscopy measures the absorption of infrared light by molecules, providing information about the functional groups and chemical bonding. FTIR can be utilized for the qualitative analysis and identification of various organic compounds in alfalfa. Proteins. Alfalfa is rich in proteins containing all essential amino acids required by the human body. These proteins contribute to the muscle growth, tissue repair, enzyme production, and various physiological functions. Vitamins. Alfalfa is a good source of vitamins, including vitamin A, C, E, K, and some B vitamins such as thiamine, riboflavin, and folate. These vitamins play a vital role in energy metabolism, immune function, antioxidant activity, and the maintenance of the healthy skin via the blood cells. Minerals. Alfalfa contains essential minerals like calcium, magnesium, potassium, phosphorus, iron, zinc. These minerals are necessary for the maintaining healthy bones and teeth, supporting muscle function, regulating fluid balance, and facilitating various enzymes reactions in the body. Fiber. Alfalfa is a good source of dietary fiber, both soluble and insoluble. Fiber promotes healthy digestion, aids in maintaining regular bowel movements, and support overall gut health. It also helps in managing blood sugar levels, lowering cholesterol, and promoting safety. Traditional use of alfalfa in herbal medicine. Nutritional support. Alfalfa is often considered a nutritive herb due to its high content of vitamins, minerals, and proteins. It has been used to support overall nutrition and promote general health and well-being. Digestive health. Alfalfa has been employed to support the digestive health. It is believed to have a mild laxative properties and has been used traditionally to aid in the digestion, alleviate indigestion, and promote regular bowel movements. Diuretic properties. Traditionally, alfalfa has been used as diuretic, helping to increase the urine production and promote the elimination of waste and excessive fluids from the body. It has been unemployed to support kidney health and address conditions such as water retention and urinary tract infections. Detoxification. Alfalfa is often included in herbal detoxification and cleansing programs. It is believed to help eliminate toxins from the body and support liver function, aiding in the removal of the harmful substances and promoting overall detoxification. Joint health. In traditional herbal medicine, alfalfa has been used to support the joint health and relieve symptoms of arthritis and other inflammatory conditions. It is believed to possess anti inflammatory properties that may help reduce pain, swelling, and stiffness. Hormonal imbalance. Alfalfa contains phytoestrogens such as isoflavones, which have estrogenic activity. Traditional herbalists have been used alfalfa to support hormonal balance, especially during menopause, and to alleviate symptoms associated with the hormonal fluctuations. Well, that's the end of my conversation, and here are some reference books. If you want any other information about my topic, you can get from these reference books, and you can get the data and all type of information from here. So thank you for your precious time. Do subscribe the channel. Thank you so much. Allah Hafiz.